Procedure to determine the effect of adrenaline on rat uterus. There are several steps, of which the first one is the preparation of physiological salt solution. Next is the arrangement of instrument and adjustment of water bath. Then balance the level, collection of required tissue attachment to the organ bath, prepare the standard drug by serial dilution and observe tissue response at different doses. Preparation of physiological salt solution. At first prepare the artificial physiological salt solution in order to keep the experiment tissue alive. The solution provides the same constituents as that of tissue fluid. The solution is isotonic with the body fluid. The physiological salt solutions are frog ringer, Krebs, Tyrode, ringer lox and Ajalon. There are variations in the composition of sodium chloride, potassium chloride, magnesium chloride, sodium phosphate, calcium chloride and glucose. Frog ringer solution is used in amphibian tissue preparation, whereas Krebs solution is used in mammalian or avian skeletal muscle preparation. Tyrode solution is suitable for intestine preparation. When you want to use experiment on heart muscle, then you have to use ringer lox solution. Finally, in case of rat uterus preparation, Dajalon solution is used. Arrangement of instrument and water bath adjustment. We have to use kymograph. The kymograph has the following components: lift screw, drum, motor box, outer lever, gear and on-off switch. The paper is used to record the tissue response. Adhesive is used to attach the paper with kymograph drum. Smoke the drum with fire which convert it to black color. There are two baths, outer and inner bath. The outer bath is a glass container that contains water to maintain temperature, whereas the inner bath is also called isolated organ bath and placed within the outer bath. The experimental tissue is hooked in it. Tissue holder with air supply are attached to the bath. Heating rod is also present. Isolated organ bath contains frontal point writing lever, thermometer, rubber tube, aeration tube, tissue holder. Lever is a device by which isolated tissue response is recorded and magnified. It has fulcrum and stylus. For magnification, lesser the inherent contractility, higher the magnification needed. This is tissue attachment point. 2 points are important: the distance from fulcrum to writing end and the distance from fulcrum to tissue attachment point. In case of rat uterus it is 4 to 6 times. For load or tension adjustment, balancing plasticine is important. For rat uterus the tension will be 1 gram. For tissue selection, sacrifice the animal by cervical dislocation. Identify the tissue and isolate it. Carefully dissect and separate unwanted tissue. Then keep the tissue in physiological salt solution. Avoid excessive handling of tissue. Attach one end of the tissue to the tissue holder and the other end to the lever. Aeration of the tissue with 95% oxygen and 5% carbon dioxide. The temperature is maintained at 37 degrees centigrade. Prepare the serial dilution of the standard drug. To observe the tissue response, pour adrenaline to the inner bath. Usually start with the lowest concentration. Observe the response for 10 minutes using the kymograph. Wash out the inner bath. Then pour another strength of adrenaline. The kymographic recording shows the pattern of contraction on non-pregnant uterus. Addition of adrenaline shows inhibition of contraction, which is recovered rapidly by Dajalon solution. But the pattern of contraction is different in pregnant uterus. Addition of adrenaline shows rapid contraction instead of relaxation.